Hello students, I hope that all of you are doing good. So today in this video lecture, we would be going through the revision of the unit 2 of the CUET biology entrance, in which we would be covering the important concepts of the three chapters, that is chapter 5, 6 and 7. So now let us start with this video lecture. So laws of inheritance. So this is very important in the chapter 5, that is the principles of inheritance and variation. So this laws of inheritance was given by whom? So it was given by the father of genetics, that is Gregor Mendel. So Gregor Mendel, after doing experiments on the pea plant, he found that there are three laws of inheritance. So let us now go through these three laws. So first law, it is the law of dominance, in which he told that the characters, they are controlled by the discrete units that are known as factors. So these factors are nothing but they are the alleles. So factors, they occur in pairs. So we know that the alleles, they can occur in the homozygous form, that is like this, and it can also occur in the heterozygous form, right? So in a dissimilar pair of factors, one member of the pair, it dominates the other. So in the heterozygous condition, like here we can see that this is a heterozygous pair of alleles. So what is happening here is that the capital letter of the genotype, it is always the dominant allele, and the small letter, it is indicating the recessive allele. So always in this pair of alleles, in the heterozygous pair of alleles, the dominant one, it would be expressed, and the recessive one, it would be suppressed. That it would not be the recessive one, would not be expressed. So this was all about the law of dominance. So the second law of the inheritance was the law of segregation. So in this, he told that during the formation of the gametes, the factors that are known as the alleles of a character pair, they present in parents, segregate from each other such that a gamete receives only one of the two factors. So only one of the two factors it would be received. So that was known as the law of segregation, and this law, it is also known as the law of purity of gametes. So this is very important. A question can come that which law is known as the law of purity of gametes. So the correct answer, it would be law of segregation. And this law of segregation, it was explained with the help of the monohybrid cross, in which the parents, they had a single pair of contrasting trait. So the third law, it was the law of independent assortment, in which Mendel told that when two pairs of traits are combined in a hybrid, so the segregation of one pair of characters, it is independent of the other pair of characters. So this law of independent assortment, it could be proved with the help of the dihybrid cross, in which the parents, they had two pair of contrasting traits. So now let us move to the next key concept. So incomplete dominance versus codominance. So this is a very important concept from this chapter, and now let us understand this. So codominance. So codominance, the name itself suggests that both the alleles, they are expressed equally. So here we can see the flower color. It has both equal amounts of red and white. So a better way to understand the codominance is by using the AB blood group, right? So AB blood group, it is the example of codominance. So in the blood group system in human beings, the I gene, it has three alleles, right? IA, IB and I. So I gene, it has three alleles, that it codes for three alleles, that is IA, IB and I. So what happens is that if this IA, it is crossed with the small I, So we have the IA itself, that is the A blood group. When IB it is crossed with I, then we have this IB and we have this B blood group. So from this we can understand that both IA and IB, they are dominant over this I allele. Okay. From this we can understand that both IA, IB, it is dominant over the I allele. But what happens in the case when IA allele it is crossed with IB allele? So we get this IAB. So both they are equally dominant. A and B, when they are fused with each other, they are equally dominant. So the blood group that we get, it is AB blood group. That is because both A and B are equally dominant. Thus we get the AB blood group. So this AB blood group, it is an example of this codominance. So now let us move ahead with the incomplete dominance. So what happens in the case of incomplete dominance? So in the case of incomplete dominance, the alleles, they blend with each other. By blending, it means that they get mixed, right? So the red flowers and the white flowers, when they are breeded, so what is happening is that we are getting a pink flower. So that is the intermediate color of the red and white. So what is happening in the incomplete dominance? Let us understand with the help of the example of dog flower. So what is happening in the case of incomplete dominance is that the red colored flower, it is fused with the white color one, right? So in the F1 generation, we get the intermediate color type of flower, that is pink. So what happens in the second generation is that it is self-breeded. So now let us make a Punnett square. So now let us write the phenotype and the genotype ratios. So the phenotype, it is the physical expression, and the genotype, it is the genetic constitution, right? So let us write the phenotype ratio and the genotypic ratio, right? So the genotypes we have are so it is 1:2:1, right? Whereas the phenotypic ratio, it is the capital R, it is representing the red color, we have the pink color, and we have the white color, right? So red it is 1, pink it is 2, and white it is 1. So in the case of the incomplete dominance, the main feature, it is that the phenotypic ratio and the genotypic ratio, it is same, that is 1:2:1, and this is seen in the dog flower. So now let us move ahead to the next key concept. So polygenic inheritance. What does it mean by polygenic inheritance? So the traits that are controlled by 2 or more genes, they are known as the polygenic traits. So they are controlled by 2 or more genes. So in a polygenic trait, the phenotype, it reflects the contribution of each allele. So the effect of each allele, it is additive. So this is a very important property of the polygenic inheritance, that the each allele, it is additive. So for example, skin color in human beings. So now let us understand this in a better manner. So what happens is that, for suppose the genotype ABC, all these three, they are representing the dark skin color in the human beings. So similarly, the genotypes that are homozygous recessive, they indicate the light skin color in the humans, and the intermediate one, it is representing a intermediate color. So we can see that here the three genes, they are contributing towards the skin color in the human beings. So now this is what is meant by the polygenic inheritance. So now let's move ahead with the next concept. So pleiotropy, this is a very important concept in this chapter. Now let us understand this. So the usual case, that is the effect of a gene, it is happening on a single phenotype or a trait. A single gene, it influences a single phenotype or trait. That is the usual manner. But what happens in the case of pleiotropy is that the single gene, it can exhibit multiple phenotypic expression. That a single gene, it is controlling multiple phenotypic expression. So such a gene, it is known as pleiotropic gene, and this phenomenon, it is known as the pleiotropy. So in this diagram, it is shown that there is a single gene here, and this here we have the beta globin gene mutation, it is present. That is in the case of sickle cell anemia. So what happens is that this mutation in a single gene, here it is causing three types of physical multiple phenotypic traits it is causing. So this single mutation, it is leading to blindness, it is also leading to liver failure, and it is also causing heart attack. So this is an example of pleiotropy. So now let us move ahead to the next concept. So mutation, what does it mean by mutation? So alteration of DNA sequences, and consequently that results in the changes of the genotype and phenotypic expression, it is known as the mutation. So chromosomal abnormalities or aberrations, what does it mean by that? So loss, that is deletion, or gain, that is insertion or duplication of segment of DNA that results in the alteration of chromosomes. So point mutation, and what is the difference between the point mutation and frame shift mutation? So this point mutation, it arises due to the change in a single base pair of DNA, and the example for this is sickle cell anemia. So what happens in the case of point mutation is that, so let us take an example of the point mutation that is happening in the sickle cell anemia. So what is happening is that in the beta globin gene, so in the 6th position, so the codon, that is GAG, it is getting converted into GUG. So there is a point mutation here. The adenine, it is getting converted into uracil because of the point mutation. So this GAG that was coding for the glutamic acid, it is now getting converted into valine. So in the normal case, the glutamic acid needed to be formed, but because of the point mutation that is happening, the adenine, it is getting converted to uracil, and the patients that are having the sickle cell anemia, the valine, it is getting produced. So it leads to this sickle cell like RBCs, that is the sickle cell anemia. So now let us go back to the presentation. So the frame shift mutation, what is happening in the frame shift mutation is that the deletion and insertion of the base pairs in the DNA, it leads to a shift in the frame. So that is what is known as the frame shift mutation. So now let us move ahead to the next concept. So Mendelian disorders. So in Mendelian disorders, what is happening is that the mutation, it is present in a single gene itself. So all these diseases, they are the example of the Mendelian disorders, and this from this a short question will come, and this is a very important slide. So color blindness, it is a X-linked recessive disorder. That is, what is happening in the case of X-linked recessive disorder is that the females will have very less chances of having a color blindness. That is because the females, they have the genetic constitution, it is XX, whereas in males it is XY. So if in the case of the X-linked recessive disorder, both the X chromosomes need to be mutated for the female to become colorblind. Whereas in the case of males, only a single X chromosome, they have a single chromosome, and if they have that single X chromosome, it is mutated, then surely they would be colorblind. So that is why the females, they have a very low chance of having color blindness. So the next disease, it is hemophilia. So also in the case of color blindness, the affected individual, they cannot differentiate between the red and the green colors. So in the case of hemophilia, what is happening is that the protein that is related in the clotting of blood, it is getting mutated, and thus the clotting of blood, it is affected. So for a normal person, if he has a cut, so the blood, it gets clotted very easily. But in the case of those patients, those who have hemophilia, the clotting of blood, it takes time, and thus it is getting affected. So sickle cell anemia, we have discussed it. So the point mutation, it is happening where A, it is getting converted into U, that causes the GAG, that is making the glutamic acid. Because of the point mutation, it is forming the valine that leads to this disorder. So in the case of phenylketonuria, what is happening is that there is an enzyme that is lacking, that converts the amino acid phenylalanine into tyrosine. So now, because the phenylalanine, it cannot be made into tyrosine, the amount of phenylalanine, it gets increased in those patients that have phenylketonuria, and it can lead to mental retardation and various other abnormalities. So thalassemia, it is a Mendelian disorder in which the body, it makes less hemoglobin than normal. So now let us move to the next key concept. So chromosomal disorders. So this chromosomal disorders, it is caused due to the absence or excess or abnormal arrangement of one or more chromosomes. So aneuploidy and polyploidy, let us understand the difference between them. So aneuploidy, it means that there is gain or loss of chromosome. Whereas polyploidy, it indicates that there are more than two complete sets of chromosome. So complete set of chromosome is there in the case of polyploidy, whereas in the case of aneuploidy, there can be a gain or loss of a chromosome. So the diseases. So this is very important, and surely a question will be coming from this topic. That is the diseases. So study it in a very proper manner. So Down syndrome, in Down syndrome, what is happening is that there is an additional copy of the chromosome number 21. So that is why it is known as trisomy 21. So in the case of Klinefelter syndrome, there is an additional copy of the X chromosome. That is the karyotype of this disease. It is 47XXY, and it is seen in the males. In the Turner syndrome, what is happening is that there is absence of one of the X chromosome. So the karyotype, it is 45XO, and it is seen in the females. So now let us move ahead to the next concept. So now we have covered all the important concepts from the chapter 5. Now let us start with the important concepts from the chapter 6, that is molecular basis of inheritance. So here there is a structure of a nucleotide. So these nucleotides, the many nucleotides, they together, they make the DNA. So DNA, it is a long chain of polydeoxyribonucleotides, right? So this nucleotide, it is made up of a sugar, it is made up of a nitrogenous base and a phosphate group. So the sugar, it is binded with this nitrogenous base with a bond. So this bond, it is known as the N-glycosidic bond. So the sugar, it is connected with the nitrogenous base with the help of the N-glycosidic bond. So the phosphate group, it is connected with the phosphate group of the next nucleotide with the help of the bond that is known as the phosphodiester bond. So this base, it is connected with the next base of the nucleotide with the help of the hydrogen bonds. So these bonds, it is very important as questions can come from this. So now let us move ahead to the next concept. So now let us understand the double helix structure of DNA. So who gave the double helix structure of DNA? It was James Watson and Francis Crick. So Watson, it is also known as Watson and Crick model. So in this, the pitch of the helix, it is measured to be of 3.4 nanometer. So what is this pitch of the helix? So it is the height of the one turn of DNA. So here this is the height that is represented by the pitch of one turn of the DNA. So this till here, from here, it is representing one turn of the DNA. So one turn of DNA, it consists of the 10 base pairs. So this one turn of DNA, it is consisting of the 10 base pairs. So the distance between the adjacent base pairs in this DNA double helix model, it is 0.34 nanometer. So now let us move ahead to the next concept. So it is very important now to discuss the salient features of the double helix model of DNA. So the first main feature of the double helix model of the DNA, it is that it is both the strands of the DNA, they are arranged in a right-handed coiled manner. So the second salient feature is that both the strands of the DNA, they are anti-parallel to each other. So by anti-parallel, it means that if one strand, it is like this, 5 prime to 3 prime, the other strand, it would be 3 prime to 5 prime. So they are anti-parallel to each other. Also, the nitrogenous bases, they are divided into purines and pyrimidines. So pyrimidines, it consists of the cytosine and thymine, whereas purines, they consist of the adenine and guanine. So in the case of the RNA, in place of thymine, we would be having the pyrimidine as uracil. So that is in the case of RNA. So RNA, it doesn't consist of the thymine. So this is very important point, that RNA, it doesn't have thymine, but it has the pyrimidine uracil. So these are the nitrogenous bases that are present in the DNA. So we can get confused that which is the purine and which is the pyrimidine. So a simple trick, it can help us to understand this. Like pyrimidines, we can remember it as CT. That pyrimidines, they are represented by CT, and C here stands for cytosine and T, it is standing here for thymine. So the other two automatically, they would become the purines. So now another important feature is that the adenine, it is pairing with the thymine, and the cytosine it base pairs with the guanine. So the adenine it base pairs with the thymine, and cytosine it is getting base paired with the guanine. So adenine it makes 2 hydrogen bonds and it pairs with thymine, whereas cytosine it makes 3 hydrogen bonds, it is getting paired with the guanine. So now let us understand what is the major group and what is the minor group. So the sugar phosphate, both of them they are known as the backbone of the DNA. So this sugar phosphate backbone, it has a negative charge. So the sugar phosphate backbone, it has a negative charge. So this negative charge, this is because the DNA it has a negative charge because of the sugar phosphate backbone. So the major groove here, it is representing that the distance between the sugar phosphate backbone, it is more, whereas the sugar phosphate backbone, the distance in the minor groove, it is less. So now we have discussed all the important points of the double helix model. So now let us move to the next concept. So packaging of the DNA helix. So in eukaryotes, the basic proteins, they are known as the histones. So the histones, they are rich in the basic amino acids that are lysine and arginine. So this lysine and arginine, they are positively charged, right? So that is why the histones, they are positively charged. So 8 histones, they form a histone octamer. So 8 histones, they come together and they form a histone octamer, and the negatively charged DNA, it is wrapped around this octamer, and this leads to the formation of the nucleosome. So nucleosome is nothing but the 8 histones that form the histone octamer. They are positive, and the DNA that is negatively charged is wrapping around this histone octamer. So nucleosome, they constitute the repeating unit to form the chromatin. So nucleosomes, they consist of the repeating units that leads to the formation of the chromatin, that are the thread-like stained bodies. So nucleosome in chromatin, they are known as the beads on the string. So here on the right side, we can see the structure of the nucleosome. So here we have the DNA that is wrapped around this histone octamer. So here it consists of the 8 histones. So now let us move ahead to the next concept. So now the chromatin, it is of 2 types. Now let us understand the difference between them. So this is very important. Our question can surely come from this slide. So let us understand. So the 2 types of the chromatin, it is the euchromatin and the heterochromatin. So the euchromatin, it is loosely packed, whereas the heterochromatin, it is densely packed. So euchromatin, it stains light, whereas the heterochromatin, it is staining dark. So euchromatin, it is, this is very important, that it is transcriptionally active, whereas heterochromatin, it is transcriptionally inactive. So here we can see that the euchromatin, it is loosely attached here, and here we can see that the heterochromatin, they are very tightly packed with each other. So now let us move ahead to the next concept. So now let us understand the experiment that was conducted by Frederick Griffith, and that was known as the transforming principle experiment. So this chapter, it has many experiments. So we need to know all the important concepts that are related to this experiment. So I would be explaining all the experiments from this chapter, starting from this. So the Streptococcus pneumonia, that is related to causing the pneumonia disease, it has 2 type of strains. So one strain, it is the smooth strain. So this smooth strain, it is considered to be virulent. So by virulent, it means that it can cause pneumonia, and it can make a mucus coat also. Whereas there is another strain that is known as the rough strain or the R strain, that is non-virulent. So there is no mucus coat that is formed by this rough strain. So these rough strain streptococcus, they do not cause pneumonia, whereas the S strain, it causes. So what is happening is that in the first case, what is happening is that the living S strain, it is getting injected into the bacteria. So we know that the S strain, it can cause pneumonia, and it thus leads to the death of the mice here. Whereas in the second case, what is happening is that the R strain, the live R strain, it is getting injected into the mouse. But we can see that after the injecting of the R strain, the mouse, it is living. That is because the R strain, it does not cause any type of disease. So what is happening is that the S strain bacteria, then it is heat killed. By using the method of heat, we are killing the S strain bacteria. So then the S strain, when it is getting injected into the mouse, as it is now being killed, so the mouse, it lives in that case. So in the last case, what is happening is that we are mixing the heat killed S strain bacteria with the R strain. So when the heat killed S strain bacteria, it is mixed with the R strain bacteria. So we can see that the mouse, it is dying. So now a question arises that why does the mice die? So after this experiment, there were other scientists who conducted many more experiments, and it was found that the DNA, it was responsible. The DNA of the heat killed S strain bacteria was responsible for transforming the R strain bacteria into virulent one, and thus that led to the death of the mice. So now let's move ahead to the next concept. So the genetic material, it is DNA, it was proved with the help of the experiment that was conducted by Hershey and Chase. So what happened in this experiment? Let us go through it. So what they did was that they took the bacteriophages. So what is happening is that we know that the protein, they consist of the sulfur, and the DNA, it consists of the phosphorus. So the bacteriophage, they were some bacteriophage, they were made to grow in the radioactive layer. They were labeled with the help of the radioactive labeled protein, and some bacteriophage, they were labeled with the radioactive phosphorus. So what happened is that they were made to infect the E. coli cells, right? They were made to infect the E. coli. So what happened is that the as the protein, it consists of the sulfur, so the protein, it was radioactively labeled with the sulfur, whereas DNA, it consists of the phosphorus, it was labeled with the radioactive phosphorus. So what happened is that when they were infected to the E. coli cells, so what happened in the last step, that is in the centrifugation case, there was no radioactivity detected in the cells. Whereas in the bacteriophages that were labeled with the radioactive DNA that infected the E. coli cells, the radioactivity was detected in the E. coli cells. So that indicated that the genetic material of the bacteriophage that was transferred to the E. coli cells, the one which was radioactively labeled with the phosphorus, that is the DNA, the radioactivity was detected there, but here no radioactivity was detected. Hence here we were able to find that the genetic material that is being transferred, it is the DNA, right? So now let us move ahead to the next concept. So the DNA replication. So the DNA it replicates semi-conservatively. It was proved with the help of this Michelson and Stahl experiment. So what they did in this experiment, now let us understand this. So semi-conservative replication of DNA, it was proved with the help of the Michelson and Stahl experiment. So what they did do is that in this experiment, they cultured the E. coli cells in the ammonium chloride medium. So they labeled them with the heavy isotope, that is the 15N. So 15N, it represents the heavy isotope of nitrogen. So that is the heavy isotope. So they labeled the first group of the E. coli cells, first group, with the heavy isotope, that is 15N. So the second group of the E. coli cells, they were labeled with the light isotope of nitrogen, that is 14N. So that became the second group of E. coli. So now what they did was that this E. coli that was, so the E. coli that was made to grow in the heavy isotope. So this was the both the strands of the E. coli DNA, they were inculcated with the heavy strands. So they were heavy. So this E. coli, then it was cultured on the 14N isotope of nitrogen, that is the light one. So what happened is that this is the first generation. So one generation of the E. coli, it takes place in 20 minutes. So E. coli, it takes 20 minutes to produce one generation of the E. coli cells. So here we have the parent heavy strands. So this is the heavy strand here, that is represented by the 15N, and this is also 15N. So now as they were made to grow in the light isotope, so they were made to grow in the light isotope, that was the light isotope of nitrogen, that was the 14N. So now it was observed that the light strand of the DNA was also produced, that was 14N, that were the new strands. So from this we were able to understand that DNA, it is able to replicate semi-conservatively. That is, it has the original parental strand, also it has a new strand, that is the light strand. So then they were further cultured in the light isotope of nitrogen, and then what it was found that we had the two, so we have here the two original parent strands. So as it is grown in the light isotope of nitrogen, so it will lead to the formation of the light isotopes itself, light strand itself. So here we also have the parental strand of the heavy isotope of nitrogen. Here we have the two strands also of the heavy strands, right? So we have the two strands, and because it is grown in the light isotope of nitrogen, the new strands that would be formed, it would be of the light strands, right? So in the second generation, that is after 40 minutes, we were able to find that there is equal proportion of the light strand and the hybrid strands. So hybrid, that consists of the both the light strand and the heavy strand, and the light strands, they were equal in the second generation. So this experiment, it proved that DNA has a semi-conservative mode of replication. So now let us move ahead to the next concept. So machinery and enzymes of the DNA replication. So DNA replication, it is very important, and the main important concept from this is that the machinery and their definition, it can be asked here. So we have the leading strand. So here we can see that the continuous synthesis of the strand from the 5' to the 3' direction, it is happening, and the strand that is synthesized continuously. So the main keyword, it is continuous, that is known as the leading strand, whereas the lagging strand, it has a discontinuous mode of being formed. The synthesis, it is happening in the 5' to 3' direction, it is in a discontinuous manner. So the keyword for the lagging strand, it is the discontinuous synthesis, and these fragments that are produced, they are known as the Okazaki fragments. So the fragments of DNA that are produced in the lagging strands, they are known as the Okazaki fragments. The Okazaki fragments, now they need to be joined to each other. How is that possible? So it is possible with the help of the enzyme that is named as the DNA ligase. So DNA polymerase, it is responsible for the addition of the nucleotides. So here the nucleotides, they need to be added. So that is done with the help of this DNA polymerase enzyme. So also it keeps the check that only a right base it is added. So if a new base, if a wrong base it is being introduced, then the DNA polymerase, it is removing that. So it has the ability to do the proofreading, that is the process of checking. So here the diagram of replicating fork, it is given. So this diagram, it is very important. So now let us move to the next concept. So transcription. So this is also a very important concept. It is the process of copying the genetic information from one strand of the DNA into the RNA. So what is happening is that this is the transcription machinery here. So here there are certain sequences that are known as the promoter sequences, in which from where the transcription, it is getting initiated, and here we have the structural gene. So these structural genes, they are usually expressed, right? So here, then after that, we have here the terminator sequences that leads to the stopping of this transcription process. So the two strands of DNA, we have the 3' to 5' strand. It is known as the template strand. It is known as the template strand because suppose the sequence of the DNA, it is like this. So the RNA that would be formed from this template strand, it would be the same as it is, but in the case of thymine, we would be having uracil. So that is why it is known as the template strand. Whereas then we have the coding strand. So the coding strand, it is from the 5' to 3'. So the another irony is that the coding strand, as the name suggests, you might think that it's coding for something, but it doesn't code for anything. So the coding strand, it is the 5' to 3', and the template strand, it is the 3' to 5'. So now let us move ahead to the next concept. So there is a difference in between the transcription of process in bacteria and the process of transcription in the eukaryotes. So now let us try to understand. The first one, we would be going through the transcription process that is happening in the case of the bacteria. So what is happening is that there are three main steps in the transcription process in bacteria. First one, it is the initiation. Second one, it is the elongation. The third one, it is the termination. So what is happening is that, first of all, in the initiation, the sigma factor So this is very important. So the sigma factor, it is getting binded to the promoter sequences in the strands of the DNA. So then what is happening is that the RNA polymerase is also getting binded to this, and a complex it is formed. So here we can see that a complex it is being formed. So the sigma factor, it is able to recognize the promoter sequence here. So then what is happening is that this together it is made as a helicase. So helicase, it has a property here of the unwinding of the two strands. So here we can see that there is a gap in between the two strands of DNA in the prokaryotes. So here this complex, it is acting as a helicase and it is helping in the unwinding of the DNA. So what happens is that now this RNA polymerase, it has the property that it starts making the RNA here, like we can see here. So now as this RNA polymerase, it is traveling further, here now we can see that it has come to the end and it gets encountered with the Rho factor. So as the RNA polymerase, it is getting associated with the Rho factor, the process of the transcription, it is getting terminated and we are getting the RNA. So in the case of eukaryotes, we do not have this sigma factor or the Rho factor. So what is happening in the case of the eukaryotes transcription process? Now let us go through it. So in the case of eukaryotes, what is happening is that the RNA polymerase, as it is getting associated with the promoter, the transcriptions, it gets start. So the transcription as it is getting started, and the RNA polymerase, then it is traveling and then it encounters the terminator sequences and it leads to the formation of the RNA. So this type of transcription, it is possible for the rRNA and for the tRNA. Whereas in the case of mRNA, there is a difference. So now let us understand the difference that is in the case of the mRNA. So for the rRNA, we have the polymerase that is taking place in the transcription. It is the RNA polymerase 1, whereas tRNA, it is RNA polymerase 3. But for the transcription that is required to make the mRNA, it is RNA polymerase 2. So we have 3 RNA polymerases in the case of the eukaryotes. So what happens in the mRNA is that we get, after the RNA polymerase, it is going through the template strand and it makes a copy of the RNA. So what is happening is, now let us go through it. So the RNA polymerase, as it is making the, in the case of the so mRNA, it is a special case. So the RNA polymerase 2, as it is getting involved in the transcription process, like in the previous slide we had learned that the RNA is getting formed. So the RNA that is formed, it is known as the hnRNA. So the hnRNA, the full form, it is heterogeneous nuclear RNA. So that is what is known as the hnRNA. So this hnRNA, it is shown here. So this is the hnRNA. So what is happening is that in the previous slide we had gone through all the process of initiation, elongation and termination. So in the case of the RNA polymerase 2, that is making the mRNA. So before this mRNA, it is the final mRNA, it is formed, the hnRNA, it is made. So this is the hnRNA here that we have. So this needs to undergo certain post-transcriptional modifications so that the final mRNA, it is formed. So this post-transcriptional modifications, they are very important for the exam and you need to know that. So now let us study that. So the first process that is happening in the case of the post-transcriptional modification, it is capping. So what happens is that a 5' methyl guanosine triphosphate, it is getting attached at the 5' end of the hnRNA. So that is the process of capping. So then what happens the next? So the first process, it is the capping. So after capping, what is happening is that the hnRNA, it is undergoing splicing. So in the splicing, the hnRNA, it is made up of certain known coding sequences. So the introns, they are known as the known coding sequences, whereas the exons, they are known as the coding sequences. So the introns, they do not code for anything, whereas the exons, they are the coding sequences. So there is no use of intron. So with the help of this RNA splicing method, we are removing all the introns that are present here. So this orange colored sequences, they are known as the introns. So here we are doing the splicing, and here we can see that there is no introns. All the exons, they are brought together. So that is what is meant by RNA splicing. So after this RNA splicing, we have the next post-transcriptional process, that is the polyadenylation. So what happens in the polyadenylation is that 200 to 300 adenylate residues are added to the 3' end of the hnRNA. So finally we have the messenger RNA after all these 3 post-transcriptional modifications, that is in the 5' to 3' manner, and we have the messenger RNA. So this is only in the case for the messenger RNA, whereas for the rRNA and for the tRNA, the normal process, it is happening, and there are no post-transcriptional modifications there. So an important point also to be discussed is that we have 3 types of polymerase, that is RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. So RNA polymerase 1, it codes for the rRNA, whereas RNA polymerase 2, it is responsible for coding the mRNA, and RNA polymerase 3, it codes for the tRNA. So we can remember it like this: 1, 2, 3. These are the RNA polymerases. This is RMT. So we can remember it like this: RMT. That is RNA polymerase 1, 2, and 3. They code for it respectively. So now let us move to the next concept. So what is meant by genetic code? So genetic code, it is a sequence of the nucleotides. So nucleotides are the nitrogenous bases in the mRNA that contain information for the synthesis of proteins. So that is translation. So the sequence of the 3 bases, it determines a single amino acid. It is known as the codon. So the sequence of 3 bases, they are responsible for coding a single amino acid. So that is what is meant by codon. So in total there are 64 codons, in which the 61 codons, they code for the amino acids. But what do the remaining 3 codons do? So the remaining 3 codons, that is the UAA, UAG, and UGA, they do not code for any amino acid, and these codons, they are known as the stop codons, as these codons, they are involved in the termination process. So they are also known as the nonsense codons. So you need to remember all these 3 stop codons, that is UAA, UAG, and UGA. So the codon AUG has dual functions. So it is responsible for coding of the amino acid methionine, and also it is acting as the initiator codon. So amino acid, it is coded by many codons. So except AUG that is coding only for the methionine, and UGG that codes for the tryptophan. So apart from these two, all the other amino acid, it can be coded by many codons. So such codons, they are called as the degenerate codons. So here we can see that the AUG, it is acting as a initiator codon, and the triplet of the nitrogenous bases, they make together a single codon. And here we have the stop codon that are responsible for the termination process. So here UAA is present, that leads to, that is, it is acting as the stop codon or the nonsense codon. So now let us move ahead to the next concept. So there are some important salient features of the genetic code. So this is also a very important topic. Let us go through these salient features. So there is no punctuation between the adjacent codons. So they are read in a continuous manner. So the genetic code, it is universal in nature. That is, what does it mean by universal? So it means that in both bacteria and human, if there is a genetic code that is UUU, so it will code for phenylalanine in both the bacteria and the human beings. So it is non-overlapping in nature. It doesn't overlap. So it is also unambiguous and it is specific in nature. That is, one code, it specifies for only one amino acid. So now let us move to the next important concept. So tRNA structure, it is a very important concept. So the Francis Crick, he told that there is a presence of an adapter molecule that it could read, that is able to read the code, genetic code, and it has also the ability to bind to specific amino acids. So this type of RNA, it was known as the tRNA. So tRNA, it has an anticodon loop. So here we can see the anticodon loop of the tRNA. So it has the bases complementary to the code. So the amino acid receptor end, that is this end of the the tRNA, it is the amino acid receptor to which the amino acids, they are getting binded. So the tRNAs, they are specific for each amino acids. So the secondary structure of the tRNA, it is the clover leaf model of tRNA. So this is the secondary structure of the tRNA. So the actual structure of the tRNA, it is the 3D form that is like a inverted L. So the 3D form of the tRNA, it appears to be like a inverted L. So now let us move to the next concept. So translation. So what is happening in the case of translation? So translation, it refers to the process of the polymerization of the amino acids to form the polypeptide. So the amino acids they are joined together by a bond which is known as a peptide bond. So amino acids, they are activated by the presence of the ATP and they are linked to their cognate tRNA. So the process of this process, it is known as the charging of tRNA or the amino acylation of the tRNA. So now let us go through the translation process. So here we have the large and the small ribosomal subunits. So what happens is that here we have the mRNA, here we have the start codon, that is AUG, and here we have the stop codon, that is UAG. So this is the mRNA. So here we can see that tRNA, it is carrying its first amino acid. So the amino acids, it is attached to the amino acid acceptor end of the tRNA. So what happens is that this UAC code, this tRNA that is carrying the first amino acid. Here, this in the process of the translation, the large and small subunits, they here bind to each other, and here the tRNA, it is getting attached to the initiator codon, that is the AUG. So what happens is that the next process, the elongation process, it is happening. So here we can see that one by one more amino acids, it is being brought by the tRNAs to the mRNA that is here, and one by one we can see that the polypeptide chain, it is growing. So in the termination process, this ribosomal unit that is present here, it reaches the stop codon, that is UAG. So it indicates that now it is time to terminate this process of translation. So thus the release factors, it is released, and we can see that the after termination, the completed polypeptide, it is being released. So these amino acids, they are attached to one another by the bond that is known as the peptide bond. So now we have completed the process of the translation. So now let us move ahead to the next concept. So we have studied all the steps of the central dogma, right? So in the central dogma, the DNA first, it is getting converted into the mRNA, and then it is converted into the protein, right? So that is the central dogma. So DNA, it has the ability for replication also. So we have studied this replication process. Then after that, we have also gone through the process of transcription by which the mRNA, it is being made. So this mRNA, this is made into, finally it is expressed into the protein by the process of translation. So we have studied all these topics, and this, it is known as, this process, it is known as the central dogma. So this flowchart, it is very important, and it can be asked in the arrange in chronological type of questions in the exam. So now let us move ahead with our concepts in the slides. So now this, from this topic, that is of the lac operon, short one question would be there in the exam. So what is happening is that, what is meant by operon? Let us first of all understand that. So all genes that are regulating a metabolic reaction, it is known as the operon. So lac operon, it controls the lactose metabolism. So here we can see the diagram of the lac operon. So here in the first scenario, there is the absence of the inducer. So the inducer, it is nothing but the lactose. So lactose, it is acting as a inducer. So here we have the Ig. So this I, it is not. So don't get confused that it is I, so it would be the inducer. No, the inducer, it is the lactose. So here the I gene, it is the regulatory gene. So this I gene, it is responsible for producing the repressor. So what happens in the case of the absence of lactose is that this repressor, it gets bind with the operator here. Here this P, it is representing the promoter. So now this repressor, as it is getting binded to the operator, so it prevents the process of the transcription because it doesn't enable the RNA polymerase to move forward. So these structural genes, then they are not expressed because the RNA polymerase, it cannot transcribe because of this repressor. So what happens in the case of the presence of inducer, that is lactose? So if lactose is present, what is happening? Now let us try to understand this. The regulatory I gene, it is producing the repressor, but here we have the inducer. So the inducer is the lactose. So the lactose, it gets binded to the repressor protein that is being produced, and it doesn't allow it to bind with the operator region. So here the operator region, it is free, and the RNA polymerase can easily move ahead, and it can take the transcription process, it can happen, and the structural genes, they can be expressed. So the structural gene, it is expressed, and the Z gene, it leads to the formation of the beta galactosidase. The Y gene, it leads to the formation of permease, and the A gene, it leads to the formation of the trans acetylase. So this is the whole concept of the lac operon. So the regulation that is shown from this diagram, it is referred to as the negative regulation. That is regulation of the lac operon by the repressor. So here the repressor, it is stopping the RNA polymerase to move any further, and the transcription process, it is getting halted. So that is why it is known as the negative regulation by the repressor. So now let us move ahead to the next concept. So the process of DNA fingerprinting. So this DNA fingerprinting process, it was given by the scientist that is named as Alec Jeffreys. So in this process, what is happening is that it is very important in the field of forensics, and also in the case of paternity testing also. So what happens is that, let us try to understand this DNA fingerprinting process. So a crime, it has happened, and from the crime scene I am able to get a hair sample. So that airs from the hair sample, I analyze the DNA. So the DNA that I am analyzing from the hair sample that I have got from the crime site, it is here mentioned here in the C column. So what is happening is that after the gel electrophoresis, the fragments of the DNA, they are being separated here. So the fragments of the DNA, it has been shown like this. So now I think that I have a doubt on two persons, a person A and there is a person B. So these both persons, I am suspecting that they might have committed the crime. So I take the DNA of the individual A and I take the DNA from the individual B, and now here I can see the fragments of the DNA, it is forming like this. So now I match the DNA that was found from the crime site and from the two suspects to identify which one is the criminal. So now I can see that the C and the B one, it is getting matched 100%. So now I can conclude that the person B, he has committed the crime. So that is how DNA fingerprinting, it is a very important method in the field of forensics. So now let us move ahead to the next concept. So we have discussed all the important concepts from the chapter 6. Now we are trying to focus of the important concept of chapter 7, that is evolution. So first we will be discussing about the origin of life. So Oparin and Haldane, they propose that the first form of life, it would have come from pre-existing non-living organic molecules, that is RNA, protein, etc. So S.L. Miller, he created an electric discharge in a closed flask. It consisted of the following gases, that is CH4, H2, NH3 and water vapor at 800 degrees Celsius. And he observed the formation of amino acids. So S.L. Miller, he proved the Oparin and Haldane theory experimentally. So in this experiment that was conducted by S.L. Miller, the question can come from which gases were used. So these gases that were used by S.L. Miller, it is very important to remember, that is CH4, the NH3, H2O and H2. So all these four gases were used, and what he saw that was the formation of amino acids, it happened. So now let us move ahead to the next concept. So the difference in between the convergent evolution and the divergent evolution, it is very important from this chapter. So here we can see these two diagrams. So here we can see the butterfly, bat and bird. they have no winged common ancestors. So all these three, they have their own individual ancestors, but they don't have a common ancestor. But in the case of the divergent evolution, here we can see that the fox and the dog, they have a common ancestor here, that is the wolf. So the first difference is that the convergent evolution, there is a different ancestor, and in the divergent evolution, there is a common ancestor. So now the next main difference it is that all these three organisms, the bird, the butterfly and the bat, they have the wings, and the function it is similar. So here what is happening is that in the case of convergent evolution, the organisms, the function it is same, but the structure of their wings it is different. So they converge to produce the analogous structures. That is, the function it is same in the case of the convergent evolution, that is flying. Whereas the function it is same, the structure of the wings it is different, as we can see here. But all these three, the wings it is used for flying. That is the similar function. But in the case of the divergent evolution, here we can see that morphologically they are very much similar to each other, but the functions it is very different. So they diverge to produce the homologous structures. That is, the structurally, the morphologically they are quite similar, but the function it is different. So the function in the case of the divergent evolution, it is different, whereas structurally they are same. So that is the difference between the convergent and the divergent evolution. So now let us move ahead to the next concept from this chapter. So evolution by natural selection. Now let us try to understand this with the help of this example, that is of the industrial melanism, right? So what is happening is that first, in the first scenario, that is before pollution it happened, before the industries they came in, and so if the industries are not there, there will be very less pollution, because the industries, they are responsible for producing the smoke that usually pollutes the air. So before pollution, the scenario it is represented in the diagram A. So here we can see that the bark of the tree, it is covered by the white colored lichens. So the white colored lichens is there, and we have here the dark colored moth, and here we have the light colored moth. So as in the bark of the tree we have the white lichens, so the predators, they would have difficulty in seeing the white colored moth, as they would be camouflaging in the background, that is of the white lichens. But it would be very easy to spot the dark colored moth in a white background. So the dark colored moth, they perished before the industrialization, whereas the white colored moth, they were able to survive. But after pollution, that is represented by the figure B. So what happened was that the smoke was released from the industries, and the white colored lichen, because of the smoke, they were colored into the dark color. So the lichen color, it was dark. So we have the dark lichens in the bark of the tree. So it was very easy to spot the white colored moth in the dark background of the tree. Hence the white colored moth, it was perished, and it was eaten by the predators, whereas the dark colored moth, they were able to camouflage in the dark background, so they were able to survive. So this is a classical example of evolution with the help of the natural selection. Like the white colored moth, they were able to better adapt to the environmental conditions after the pollution and the industries that were set up after pollution that happened. The industries were set up. The white colored moth, they better adapted to the environmental conditions and they were naturally selected. Sorry, the dark colored moth, they were able to survive after the pollution. So the white colored moth, they were perished because they were easily seen by the predators. So after the pollution, the dark colored moths, they were able to survive and thus they better adapted to the environmental conditions, and they were able to survive because of the natural selection process. The white colored, they were perished because they were easily seen in the dark colored background. So now let us move to the next concept. So Hardy-Weinberg principle. So a numerical question, it can come from this Hardy-Weinberg principle. It is very easy. So now let us go through the points of this principle. So this principle states that the allelic frequencies in a population, they are stable and it is constant from one generation to the next. So the gene pool. So the gene pool, it includes the total genes and their alleles. In a population, it remains constant. So this is known as the genetic equilibrium. So sum total of all the allelic frequencies as considered by the Hardy-Weinberg principle, it is 1. So the equation that is given by the Hardy-Weinberg, it is p square plus 2pq plus q square, it is equals to 1. So the p square, it is the frequency of the dominant alleles. The q square, it represents the frequency of the recessive alleles in the population, and the heterozygous allele population, the frequency, it is given by the 2pq. So the change of frequency of alleles in a population, it disturbs the Hardy-Weinberg equilibrium. So now let us move ahead to the next concept. So there are three types of natural selection. So the first one, it is the directional selection. So in this, the one extreme trait, it is being selected and those organisms, they are able to survive, whereas it is against the other extremes and those that are having the median type of traits. So what is happening is that, so for example, in a population, there are some tall people. So those tall people, they would be selected and they would be surviving, whereas the short people and the medium heighted people, they would perish. So that is what is meant by directional selection. That is selection, it happens against one extreme only. So the graph, it is shown here like this. This was the original population, but as the long people, they were being selected and those survived, so that is why it shifted towards this, and the short type of people, they perished. So what happens in the case of the stabilizing selection, it is that the medium heighted, for example, in a population, the medium heighted people, they are able to survive. So here both the extremes, that is the short heighted people and the long heighted people, they both perish. So that is what is meant by stabilizing selection, that it works for the moderate traits, whereas it is against the both the extremes. Here we can see that this is the original population, whereas the population after selection, as it is favoring only those people that have a medium, that have a average height. So we can see that only the medium people, they are getting selected, whereas both the short heighted people and long heighted people, they are perishing. So that is what is meant by stabilizing selection, that both the extreme traits, they get perished. So in the case of disruptive selection, the the people, those are having the average height, so they are getting perished, whereas the short heighted people and long heighted people, they are able to survive. So the disruptive selection, it is for both the extremes. Both the extremes, they are able to survive, whereas it is against the moderate traits. The medium traits, they are getting perished. So that is what is the difference between all these three types of selection. So now let us move ahead to the next concept. So these were all the important concepts from the chapter 5, 6, 7 of the unit 2 of the CUT biology entrance. So hope that this PD revision session was beneficial for you. Thank you for your valuable time and patient listening. So we will be meeting tomorrow at the same time at 5 p.m. for the unit 3 revision.